I think we'll call time. So thank you to everyone for joining us today. So we're starting to fill the room up again, so it's good. So today our speaker is Tim Buschman. Tim is visiting us from down the road at Princeton. And so I'm currently, I've mentioned this before, I'm organizing talks this year, and so that means that I get to invite all the people that I really want to see. And at some point you guys are going to get really tired of that, so I will also invite you to please let me know if there are any speakers you would really like to have visit, and we'll be happy to organize that. But for now, I get to invite, and I saw Tim's postdoc give a talk at CCNP, so the Center for Computational Neuropsychiatry, which is the Rutgers Princeton Initiative. And, well, I just loved it, and I wanted to hear more. And so Tim is joining us today. Tim what did I just learn? Did his bachelor's at Caltech, and then did his PhD at MIT, and has been down at Princeton for coming up on a decade? Yeah. It's been a while. Okay. And is going to be talking about working memory today, but also does a lot of other really interesting things. So I'm really looking forward to this. Thank you so much.
Yeah. Thank you, Pernille, for the introduction and for the invitation. Yeah, it's been fun, and thanks for squeezing me in, like in this sort of pre-SFN days. It's always tough. So yeah, I'm going to talk a little bit today about some work that we've been doing, trying to understand kind of the neural mechanisms that underlie working memory. It's going to be kind of a combination of two different studies. Talk about something that we kind of presented at CCNP, like sort of a behavioral model of working memory, and then for the first half, and then the second half will be some newer unpublished data doing some recordings in non-human primates. So my lab does a lot of different types of approaches, and so the talk will be kind of trying to maybe bridge across a couple of these different domains. Please interrupt me if anything is unclear. Any questions come up, I'm happy to be. We were talking about this before. Princeton is like one of those places where like people jump in right away, and so I'm used to it, so feel free. Okay. So yeah, so when we think about working memory, again, I'm sure everybody here knows this, right? But we sort of think of working memory as essentially your ability to hold things in mind, right? And we think about it as this kind of flexible workspace, which you can place items into working memory, you can manipulate them, you can use them then to guide your behavior. It helps you kind of bridge from the immediate sensory world to some action at some later time point, right? So it's really kind of fundamentally important for pretty much all cognitive processes. Despite being so important, it turns out that working memory is kind of fragile, right? So it's very susceptible to errors, and it's susceptible to interference, right? And I can give you an example of this. So for example, if you've ever logged into your Gmail on your computer, right? This is like the modern day equivalent of like the classic remembering your phone number, right? Like now you have to remember these verification codes. And it's hard to do, right? It's like a random 6-digit string. Oftentimes you might transpose them. It'll take you, at least if you're like me, it takes you a couple times going back and forth to kind of get it correct. And so this is sort of reflecting the fact that first of all, working memory has this kind of capacity limit that you can't hold an infinite amount of information in working memory, usually about 3 or 4 items. And that it's susceptible to these errors, right? So like you might misremember one of the digits, or you may transpose them or something like that. And it's also susceptible to interference, right? So if you had an annoying older brother like I was, right, and somebody was trying to remember a phone number, right, like you would always just stand next to the person and just start shouting random numbers at them, right? And this is an example, right, where like sensory inputs are interfering with your ability to kind of hold something in working memory. And so we're really kind of interested in this question, right? Like what leads to these errors in working memory? How does the brain mitigate the kind of cause of these errors? And what might be some of the neural mechanisms that will protect working memory from external stimuli, new distracting stimuli? Okay, So these are the the sort of two projects I'm going to talk about today. As I said, the first one's going to be really kind of modeling of psychophysics of behavior, and then the second will be actually looking at some neural recordings that we've done in monkeys. So how are we going to look at working memory? Well, we kind of took the approach that has sort of become dominant, I think, in the field over the last 5 to 10 years. We're using a delayed estimation paradigm, right? So in this case, use my new
oh yeah.
So in this case, right. Okay. So time. This is the task that we gave to humans. So time is kind of moving from the upper left to the lower right. So they begin by fixating. They're presented with an array of colored squares. This could either be 1 or 3 colored squares that kind of appeared randomly on the screen. Then there was a memory delay in which they had to remember the location and the identity, the color identity of these squares, either for a short period, 1 second, or for a longer period, 7 seconds. And then at the end of the trial, we indicated which one of these 3 objects the human should report, right? And so we just sort of put a square there, and they used their mouse to kind of rotate this ring. And as they rotated the ring, the color here would change, and then they would kind of click to lock in what they remembered the color to be at that particular location, right? And the nice thing about these approaches is that you can kind of get a measure of error on every single trial. Right, so here the true color was, say, something like close to red, but perhaps the subject might report something that's closer to pink. And so we can use this angular difference on this color wheel as kind of like our indicator of error. Right, and we can, in fact, do something more. As I'll show you later, we can actually look at like where the error is. Did their memories drift left or did they drift right? And so if you just plot, this is just a distribution of across all of the trials that humans did. I don't remember how many subjects are here, hundreds of subjects, right? I mean, it's MTurk these days, so whatever. And so, hundreds of human subjects, if you just plot the angular error, right, which can go from -180 to 180 across all the trials. This is with when they were asked just to remember a single stimulus and only remember it for 1 second. And you see a nice distribution that's kind of tight around 0. Meaning that they tend to report the color that they were asked to remember. Okay, they're doing the task. As I was saying before, working memory has this capacity limit. So if I ask you to remember 3 things rather than 1 thing, then your memory accuracy goes down, right? And you can see this reflected in this distribution where the number of kind of perfectly reported stimuli is lower, and you get more errors out here in these tails. And also time matters, right? So as there's some process that's sort of accruing errors over time, so whether or not I ask you to remember 1 or 3 items, if I ask you to remember it for a longer period of time, so instead of 1 second, now 7 seconds, your errors again are getting, your memory's getting worse, you're getting less accurate, so your errors are getting broader. As I said, this was actually a task that we did both in monkeys and humans. Everything, looks like 90 subjects. Everything I'll be showing today is basically identical across the 2 systems, so I won't really be making a big difference out of it. The behavioral task was essentially identical for the monkeys, except they were getting this kind of abstract cue that was telling them which one to report rather than a spatial cue. And they were reporting by making an eye movement to a color wheel at the end of the trial. But otherwise, we can get exactly the same thing, right? We can get the angular error on trial-by-trial basis, and we can look at their performance as a function of load, whether or not they remember 1 or 2 items, and whether or not they were remembering it for a short or a long period. Here, again, the delays are different. It's ½ a second to 1.5 seconds rather than 1 or 7 seconds. But again, like you see almost identical results. Monkeys are a little bit worse than humans are. Part of this may be just the way that they're responding with the eyes rather than kind of like with the wheel, but there is also a lot of work showing that there's a strong correlation between working memory capacity and general fluid intelligence. So, hopefully this will make you feel better about human race. Although today is election day, so who knows? We'll have to see in a couple hours whether this holds up. All right, so what is driving these errors, right? So the classic theory of this is that it's just due to random diffusion of the memory, right? So if you have some memory, some color that you're trying to remember, this kind of red pinkish, over time, due to stochastic noise, like either due to the sort of stochasticity in the production of spiking activity in the brain, or you can think about neurotransmitter release. There's a lot of sources of kind of variability and stochasticity in the brain. And so the idea is that you're going to get this kind of diffusive process where memories are kind of just going to erode over time. And this can kind of explain a lot of these effects, right? Where the longer you're holding something in memory, well, of course, the longer this diffusive process will run. And if you have some effect of load where this diffusion happens quicker as you add more items into memory, then you can really explain this type of curve here just simply from this diffusive process alone. But when we started looking at this data a little bit more closely, we noticed that humans' reports were kind of clustered, and monkeys' reports were kind of clustered around specific colors. And so this is showing a distribution, so the probability of a human or a monkey reporting that the remembered color was at one of these specific hues as a function of hue, right? So the colors are actually shown here, right? And the target distribution that we're showing both humans and monkeys are completely flat, so it's completely uniform. Yeah?
I have a quick question. So this is actually back to like 2 slides ago. You showed the distribution and the tasks for the distribution of errors and the tasks for both monkeys and humans. And you made this point that maybe part of this has to do with the difference in monkeys versus humans. Some of that difference might have to do with the mode of response.
Yeah.
So I just want to address this because it seems like my intuition is that the effect of the mode of response would actually go in the opposite direction, right? In that I'd expect more error in the case where you're showing one color at a time, and it's sort of like there might be effects of maybe adaptation or all sorts of things because you're presenting a particular color, different biases. Versus if you had humans just make an eye movement to the correct color, one, there's less of a delay, right? You don't have to keep it in memory for quite as long because you don't have to manipulate this thing. And then, I mean, there's the calibration error part, but you're not. So one thing I'm going to want to ask later, I guess, for the human data is, do you find a dependency on where you start the color?
Yeah, so we start the color randomly, so there's no dependency. There's no that's not something we really manipulated, I should say. I don't know how systematically we looked at that. I think generally, you do find subjects will sort of under-report, right? So they'll tend to move from where they are towards but never quite go past, right? They always will undershoot rather than overshoot. And so there is probably, there's again, we haven't looked at it systematically, but my guess would be that you're going to find some biases there. But again, because the starting color is random with respect to the actual color, the hope is that across hundreds of trials per subjects and hundreds of subjects that kind of washes out, for at least for the effects that I'll show you. Although, I mean, please, if you think it may come up later, then re-raise the question. But to address your other question, so we never actually ran humans on actually doing eye movements. We did run some subjects in-house where we could control eye movements, and they had to fixate up until the response. And we ran, but a lot of our subjects run just through mTurk where we're not controlling eye movements. You get identical results. I think the difference is right here you're able to fixate specifically at this color and get good color information, right? So color drops off pretty quick. In the monkeys, we put the color wheel at kind of the 3.5 degrees, 4 degrees, and so it's still within color vision, but they're probably not getting as good, right? I think it's a fair point that we never really ran the experiment to say exactly like is this motor or not. But, yeah.
So I'm going to try and make a prediction here that it's actually related to the picture you're showing right now, where humans have more color categories. And they're biasing towards those color categories, making the error less. So when you have more categories, fewer categories, your errors by definition are going to be greater.
Sure, but it could be both, right?
Yeah.
Sure. can compound it, but just looking at this, that'll immediately
Yep.
lower your
Yeah. Yeah.
your accuracy.
Yeah, right. So I think, right, so that's sort of something I'll sort of explain where we think these peaks are coming from later. And yes, it seems like humans have a finer discrimination than monkeys do. This is something that's been reported in the literature often, and often people assume that it's due to linguistic labels, right, of like blue, green, red, and purple, I guess. But monkeys don't have language, or at least we don't think they do, right? And so, where are they getting kind of brownish, orange, and kind of reddish from? I guess this is more blue and red. I think that's, a good question. Yeah.
So I have another question quickly. So before we get too far, which is, presumably these monkeys have been trained to do other tasks.
No, actually, these monkeys were just trained to do this task. Yeah. And because I get this question a lot, monkeys and humans have, at least at the retinal level, have nearly identical distribution of photoreceptors and things like that. So I mean, people have characterized this fairly well over the years. Our monkeys weren't colorblind. Like we tested for all of this.
If you're training monkeys to do that, so obviously, right? My implication is that if you train monkeys to use colored cues in some other task, they may have. Yeah.
Yes, right. So I can give you a hand wavy explanation for where these come from later. I guess the other thing. What was I going to say? We tried real hard to train this out of them. Let me say that. Yeah, we saw this and we said, "Oh, they're cheating," and so we did everything we could to try to warp the space and get it to go away. We never could, and that's what actually kicked off. So we found this first in monkeys, and then that kicked off our study in humans. Yeah. Okay, so what might be going on to sort of lead to these? Sorry, let me finish this. I think hopefully this is clear, right? But just to drive home the point, right? So there's these sort of clustering where people and monkeys tend to respond and report certain colors more often than what you would expect from the actual distribution itself. So the question then becomes, what's driving this, right? And so our model of this is that there is this diffusive process going on, but rather than it kind of existing over a flat, uniform space across colors, instead there's kind of you can think of it as kind of hills in color space, right? Where there's certain colors that are attractive, right? Like you're going to kind of slide downhill towards the valley of these peaks, and other colors where there's an unstable attractor and some unstable fixed point, and some where there's going to be a stable fixed point, right? And it's going to pull memories into these locations. And so what we're actually proposing is that there's two forces really acting on memory. One is this diffusive force, and the second is this what we're going to call a drift force that's kind of, again, pulling you towards certain attractors. And so what we did is we developed this is all work from Matt Panicello in collaboration with Brian DiPasquale in Jonathan Pillow's lab. And so Jonathan Pillow thinks a lot about these things, and so as soon as we brought this to him, he was like, this is how you do it. And so this is how we did it. We just basically built a dynamical systems model of how memories should evolve over time, right? So we're making a function, we're going to try to fit a function that says, given the current color identity, which is going to be in the circular space, right? Color circular. So the current angle of the color is going to evolve in some way. It's going to change over time, and that change over time is going to be due to two forces. One is this diffusive random noise force, right? So this is just a random walk in space. And that's going to be, have some, as I was saying before, some variance on that is going to depend on how many items you're holding in memory. And the second is going to be these attractor dynamics, this drift force, which is going to be encapsulated in this G term. And again, there's going to be a parameter that's going to modulate the strength of these drifts as a function of say, load. Okay, so the way we fit this drift function is that we fit it as kind of a weighting of a variety of different bases that sort of span color space, and this is like an example drift that we got out of, say, one human participant, right? So this is kind of the real life example of this schematic, right, where you can kind of see these peaks and valleys, although because this is the differential, right, this is the change in angle over space, you can think of this as kind of like the differential of potential energy is going to give you this function here. And so what that means is that there's these certain points 0 where there's not going to be any change over time, right? And again, positive number here is going to mean that the change is going to push you to the right. Negative means you're pushing to the left. And so when you have these kind of negative slopes around 0, that's a stable attractor. Both sides are kind of moving towards that middle point. Okay? This is what it looks like if you kind of think about, like, here's color space here. These arrows are showing this drift function, again, for just one sample subject. And so let's just imagine one solitary trial, right? So we put in some green color. How is the memory of that green color going to evolve over time? You're going to go through some encoding process, which is also going to have similar kind of diffusive and drift effects on it. I'm not going to talk about that in a whole lot of detail, but in fact you find basically identical functions for this encoding process as you do during memory. Then after it's encoded, so at this gray point here is when this subject's holding it in memory. That, again, the memory is just going to kind of diffuse over time. It's going to drift along this gradient, kind of move down this gradient until it gets to kind of a fixed point, but there's always going to be this kind of noise that's bumping it around. And so that becomes clearer if, this beta term is going to change the magnitude of this, the kind of the arrows here, right, that are pulling you in different directions. And then, again, this sigma is telling you how noisy this process is. So if we put in a lot of trials, right, the same starting color here, you're going to get generally following a similar trend, although, again, there's going to be noise in them. Right? And some colors, if you start it at one of these kind of unstable fixed points, you can get really diverse distribution of colors. Yeah?
So do the subject ever get feedback on their color choice?
Yes.
So they know they have the correct choice or not?
So we train them ahead of time, and then during the actual trials, we don't give them feedback anymore. But early during the training period, we give them very explicit feedback. We say you're off by 10 degrees, clockwise or counterclockwise, and we sort of show the two colors and all of that.
Right, so when you model the drift with training, is it less than without training?
Oh, that's a good question. We didn't have enough training trials we never really looked at that, but I think we would not have enough training trials to really do that. But that's a good question. So if we changed the feedback, so if we always gave them feedback, would they improve? Yeah.
I've done lots of these color tests, and I don't think it makes a difference if you train people or not. There's bias in recall for color, regardless of.
Okay, because like if you're telling kids how to color something red, you say, oh, this marker is more like red. Eventually they'll pick the right crayon to epoxy.
Right.
Yeah, and that's sort of like the linguistic label sort of hypothesis.
Because it's discrete color choices rather than like holding this
Yeah.
memory kind of.
So towards the end, I'll show you, I think, an experiment that hopefully addresses a lot of these concerns. Yeah, that really. Well, I mean, first of all, again, like the monkeys don't have language, and also this final experiment where we actually manipulate the distribution of colors that they see, and that changes everything. And so I think that also will. Hopefully, but let me know if it doesn't. Okay, this is just showing that this drift and diffusion model does a better job of predicting both human performance and monkey performance than just a diffusion alone model where there is no kind of structure within color space that's kind of pulling or pushing memories in different directions. Okay, and so the nice thing about this is if you kind of run a bunch of simulated data through the colors that the human subject saw, and you kind of run this out, you get a distribution that looks very similar to the experimentally observed distribution. So it seems really like that the model's doing a good job of capturing this statistic in terms of like the subject report. Yeah.
Real quick, but so the parameters for your model that tell you where the drifts go, did you get that from these observations, or did you get that somewhere else?
So all we're doing is we're fitting the model in a way that is trying to as accurately put in a color here, and you simulate it out, and you're trying to predict, given that starting color, what will the human subject report at the end. Yeah. And so everything's just coming from the that alone. And in terms of like identifying where the attractors are, that's just coming out of that fitting process. Yeah.
Related question, is the number of attractors constant? And if not, how variable is it across individual subjects?
It's not constant. There is variability. Some go from 3, some go to 5. One of the monkeys had 2, one of the monkeys had 3. I think this might be I think this is just one monkey actually here, because there is some variability there. Yeah, so there does seem to be individual variability. Again, I think that the literature on these kind of like where these are is pretty rich in the human psychophysics, and there's been lots and lots of studies. I mean, I think it varies across like culture. Yeah.
But presumably not for monkeys. So that's why I was asking.
I mean, we've thought about this, yeah, and again, we have, we do have some recordings now where we've manipulated what the colors the monkeys see. It's still a little bit early days, but I can talk, if people are interested, I can talk about what we see neurally that might be driving this effect. But yeah, it has to do with like compression and expansion of color space in the cortex, in prefrontal cortex. Yeah. All right. So this is all from fitting one of these complicated models, and you may be thinking, okay, yeah, so you had 2 models, you fit one, and it fit better, but like, is this real? And so we wanted to come up with some like model-free statistics. And so, if this model is true, what are the predictions that we can make? One is that for certain colors, you should get very consistent drift. So if I put in this kind of reddish color, I expect to get an orange color out if there is an attractor here, right? And so what this should look like is a bias in the response. So every time I put in this target, I'm still going to get a noisy distribution of responses, but the mean of this is going to be biased in one particular direction. And of course, if I put a color on the other side of this attractor location, then I should expect to get the opposite bias, right? And so we can just plot this bias as a function of the target color relative to the nearest response peak. So we're going to assume that these response peaks are actually these attractors, and so things should be pulled in towards them. And so again, with just by convention, what we expect here is that positive numbers mean that you're drifting to the right, negative numbers mean you're drifting to the left, and so we should see kind of a negative slope here that kind of crosses around 0, meaning that you don't change when you're presented with a color that's actually at the response peak. And that's what we see for both humans in black and monkeys in gray. Right? So there's this really nice kind of attractive bias towards that location. Another prediction is that how much variability there is in the response should also vary as a function of where you are with respect to these attractors. And so if you're in the attractor, right, the attractor itself is kind of fighting against this diffusive process. It's anytime you move away, it's going to be pulling you back. And so you would expect your standard deviation of this response to actually be kind of smaller when you're at one of these peaks. Then when you're off peak, you actually expect a much larger diffusive force. And again, that's exactly what we see, both humans in black and monkeys in gray. Around the peaks, you have lower standard deviation in the report. And then as you move kind of away from that, you get this increase in standard deviation. So this begins to give us some idea about why you might want to have these attractors as well, right? Is that they in some way can kind of mitigate the noise. And so that's what we're going to sort of look at next.
So, I mean, let me just put this in a very crude way. There's happy colors and unhappy colors. And if you were measuring it, right now you talked about measuring on the slope, but if you actually pick the peak of the unhappiness, you should have the maximum standard deviation, and it shouldn't have bias because it'll be going in both directions.
You might still end up with a little bit of bias because it's not a completely kind of symmetric, unstable fixed point, but yeah, in general, yes, yeah. So if you replot this as a function of the troughs, right, then yeah, you see essentially the opposite, where you get the largest variance at the troughs, where people don't tend to report things. Yeah.
Could you
give me, I'm struggling, I'm trying to get an intuition for the difference between the predictions of this attractor model versus, say, you know, a model like Camilla and Pernilla have, and other models that are either hybrid, like you have a category-based representation, which you combine with the input, or you have like a prior on particular colors or something like that. And at the growth scale, it seems like you'd have these same sorts of predictions, right? You're going to tend to be biased towards whatever the nearest mode is. And so I'm trying to figure out how these predictions differ, at least qualitatively.
Yeah. In a lot of ways they don't, right? I mean, I think the one thing I will say is, depending on how you would integrate categorical information, you may not expect it to sort of evolve over time. So that's a big thing here, is that we really capture both early and late reports. So I can tell you as a function of when I stop your kind of memory process, what your error is going to be, and that's going to evolve more, and you're going to see stronger dynamics dependent on kind of how long it's sitting in working memory. Of course, you could always
same thing if you just thought that the memory trace like degrades. Right?
Degrades, and so you rely more on like your sort of prior expectation. Yeah, 100%. So the other thing is that we're excited about this because it's something that you can imagine very clearly how you might implement this in a neural network, right? And so I think that's sort of why I think it captures a lot of the same effects, but in a way that we might expect maybe some neurons to do things, right? I mean, we think about attractors in neural space, and you think about, I mean, again, you could imagine like, so there's category color neurons, and then there's, non-category color neurons, and they are both feeding into some decision circuits. Yeah, I mean, there's other ways to imagine, of course, which is why at the end of the day we do monkey work, so that we can try to maybe address this by recording and seeing what we find.
I mean, so the predictions are exactly the same for the two models. Camilla's nodding along as I'm saying this, right? But one difference too is that like our models are rational models, and these are process models. So you're operating at a slightly different level, and he's learning the parameters. And I don't know if you did one where you were actually like we did that recursive inference, right? So we're using like environmental statistics for the. But he's learning on the fly over time how the categories.
No, I understand that. I'm just saying.
So it's just I think it's a different implementation of the same idea. I don't think that there's really a substantial difference in.
I mean, I hit a slide last night that I will put back in. I'll try to.
Okay.
Well, I'll try to make this point like maybe more clear. Like that this we think about this as a possible way in which you could implement Bayesian inference in an attractive neural network, basically. Yeah.
Right. So go back one to where you have the regression lines between the, so I'm calling regression lines, but that, right?
Yeah.
So this is exactly the prediction that you would get from the Bayesian model as well, based on the difference in the strength of the priors and the noise in memory, right? And so the attractor in your model does exactly the same thing as the strength of the prior and the Bayesian.
Yeah. That's the hidden side. I think I try to make that part.
Yeah.
All right. So we're just again trying to make this place like it's like we think about this as like maybe a potentially mechanistic explanation for these things, just at a deeper level. Yeah, or more. That came out wrong. Like a more circuit-based level. Yeah. All right. So what is the advantage, right? And I think I already kind of spoiled this, right? So the idea is that diffusive is this process that kind of goes with the square root of time. The longer you're in there, your error is just going to grow infinitely long. Whereas if you have this kind of drift function, as soon as you fall into one of these, you're going to be somewhat stable, right? If you get enough noise, you can kick out and get into the next well over. But in general, it's going to kind of bend this curve down for longer times in memory. But this comes at the cost of actually causing errors early on, right? When you fall into this attractor, it's actually worse than you would have done otherwise. for these sort of short memories. And so we were curious about this, and so one idea we had is, well, again, sort of inspired by these Bayesian ideas, right, is that, well, this model here is assuming that colors actually are drawn uniformly, which they are in our task, but in the real world they're not, right? You have a lot of biases in the colors. And so what would happen if, you lived in a world where, you see lots of brown trees and green grass and blue sky, right? And so you have kind of a lot of common colors that you run into a lot. And what if these attractors were kind of lined up with these common colors? Then, if you just run a simple model, you actually do better with this sort of attractors, with those kind of attractors fit or optimized to the actual distribution that you see, then you do better than you would do even with diffusion alone, and even for these short time periods, right? So like you just tend to fall into the error you're making tends to be towards what you would want to respond anyways. Again, from like a very Bayesian perspective. And so we wanted to test this. And so the way we did this is we did this in humans, and we've subsequently done it in monkeys, which is something that we're kind of like almost done in analyzing now. But for the humans, what we did is we just, again, had them do the same task that we had before, slightly simplified, so they only ever get 2 samples. And now, rather than these colors being drawn completely uniformly, there's a bias, right, where there's certain colors, there's these 4 colors that the human subjects saw repeatedly. So they would often see purple or blue or green or red. They were always equally spaced like this, but what 4 colors were more common were completely random across subjects. So every subject had a different kind of comb of color density. And so if we think that these attractors are adapting or adaptive, right? They're intended to help your ability to do the whole things in working memory, then what you should see is that you should see attractors form at these locations. And so that's what we wanted to test. And so again, we can do this in a model-based way, or we can do it in a model-free way. We'll start with the model-free way. We'll look at the response bias, again, like how much does the mean report vary from the put-in color, or the presented color, as a function of distance from one of these common colors now, right? So this 0 here is the center of that common color spike. Early on, there's not really any kind of slope here, right? If anything, it's kind of a weak slope. So this would be, they did 600 trials, I believe, so I think this is the first 200 trials that subjects saw this. And again, this makes sense because across subjects, you tend to have fairly consistent kind of locations of these attractors. And because these kind of common colors were random, you would kind of expect that they would fall randomly with respect to the actual, like, their pre-existing attractors. And so you don't get much of an effect here, but if you now look at the last 200 trials, you get this nice attractor that's formed at the commonly presented colors. Right? And I think this, for us, kind of helped us feel okay about the fact that, again, this isn't having to do something with like just the photoreceptor distributions or their like tuning curves in the retina or anything like that, right? This is all that presumably is being fixed over the 600 milliseconds. 600 trials over 45 minutes that the subjects are doing this. We can also do it the model-based way. We can fit that full dynamical system, and we can ask like where do attractors form, again relative to the common color, and this black line here is where the attractors form, and they tend to form more at the common color than what you would expect by chance. This gray shaded region is kind of just the chance distribution. Okay. So so our argument is that these attractor dynamics may mitigate this impact of neural noise, this kind of diffusive process, and that this could, particularly when you adapt to the context, this could really improve memory performance. Let me show you the hidden slide, right? And so we, again, I don't know, I mean, I think this is cool, right? That this is maybe a way, these attractor dynamics may be a way in which you can implement these sort of Bayesian inference over time, right? So you have some sensory input, some sensory evidence that you accumulate during stimulus presentation. If you have these priors that are embedded in the dynamics of the neural network itself, then over time, what will happen is that this sensory evidence will sort of, as it's the longer it sits in working memory, the more it sort of integrates in this prior distribution that's, again, kind of encapsulated by the dynamics itself. And so we think, again, this is maybe a mechanistic way of, or a circuit level way in which you could think about implementing Bayesian inference. Okay. Any questions or more questions on this? Yeah.
When you talked about that column of representative colors that people were repeatedly presented with, were they always related to the category that they belonged to, or could come from like randomly distributed 4 different numbers? Was it like a different purple every time, let's say, or could it be only?
Yeah, they were completely random. There was always this they were always equally spaced.
Oh, equals.
Yeah,
Okay, that's
but
it.
in terms of like the, right, so in terms of like the phase offset, or kind of like what this color, this primary color, it could be anything.
Uh-huh.
Yeah.
Because my understanding, not knowing that they were equally spaced out, that would mean that maybe that would kind of get rid of the whole, environmental statistics having an importance in terms of prior story. Because if they could just change in the span of 400 trials, they're all like prior existing numbers, then that would, to me, not make sense, but not make sense.
I think, yeah, so this doesn't overwrite. So all we're doing is we're looking at how these attractors are forming near these common colors. What I'm not showing you is if you keep the old attractors, you still get this. It goes down, so it gets weaker, but it doesn't go away. And so it's not that people are completely forgetting their previous expectations. We also did an analysis, which I don't know if it actually was in the paper. So take it with a grain of salt, because maybe it didn't hold up to rigorous. But I do remember doing an analysis where we asked, okay, if you put one of these common colors kind of nearby where an attractor used to be, or you put it in like one of those bad colors, right? It takes people longer to sort of form the attractor at the bad colors than it does at the. So it's like it seemed like they kind of were nudging their expectations, but like really reforming that map definitely took more effort. Yeah.
One more quick question. Is there any conceptual limitation on essentially a discontinuous prior distribution? If there were certain colors that were impossible, any reason you couldn't just have effectively infinite attractors?
Right. I mean, yeah. It's a good question. Is there any perceptual space that is discontinuous? I guess.
But there are experiments in which certain colors are never the right answer.
It's true. Yeah. Yeah, not a manipulation we've tried, but yeah, it's interesting. Yeah. Yeah. I mean, so this is circular, right? Which is kind of it's nice in that you don't have these edges, but other spaces that you do, like say spatial frequency, you do tend to get these attractors. You tend to get convergence towards the middle. Interestingly, like orientation, you see a very similar type effects, but you would expect that your sort of predominant expectation for orientation would be vertical and horizontal. That's what you see all day long. These peaks tend to lie at 45 degrees. So I don't know what to make of that. But
So, and maybe this is a little bit of a side point. So one issue that I've had with the way that you described a lot of this. So I don't know, have people actually tried doing, I mean, these are always fraught, right? But try doing environmental measurements of like what hues the.
Oh, I don't know if I have that slide, but yes, yeah, absolutely. You can just.
But because I'm just saying, I think they're very fraught, but I think.
Yeah.
But I think it's right. The thing that really matters. So you've been framing this in terms of minimizing errors, but right, it's minimizing costs that really matter. And so I feel like in a lot of these where you're showing this, there's a plausible reason to want to discriminate between, say, red and green. That's very obviously, if you're an animal that eats fruits, for example, right? That's a categorical boundary that really matters. And so within red, like how close you are doesn't really matter.
Yeah.
I mean, I guess this matters. This is more for the story, right? But so why do you why do you develop these attractors or whatever? But my sense is that it probably has more to do with it. I mean, your experiment is purely in terms of, in that case, you're setting it up so that all errors are equally costly, I guess, right? Your fake spaces where you set up these intervals.
Yeah, for the humans they are. For the monkeys they're not.
Ah, okay.
So they do get rewarded as a function of how close they are to reproducing the correct.
Oh, sorry. By equally costly, I mean this given distance from the category matters equally across. So what matters is the distance of the squared, right? Whatever it is, the squared.
Yeah, the angular distance.
Yeah.
Yeah.
Or whatever it is, the angular
Yeah.
distance from the category. What matters is not whether you cross the category boundary or not. Right. So I'm saying in
Yeah, other than like if you're going to jump category boundaries, that tends to be a larger error than if you just drifted nearby.
Right.
Yeah.
But so just to back up a little bit, what I'm saying is, or what I'm contending, I'm saying that in real life situations, you
Mhm.
make decisions, and certain decisions, though they may be far, like you categorize this as a green,
Yeah.
but the wrong green, but in terms of consequences,
Yeah.
it doesn't really
Yeah,
matter.
it's not a huge cost.
Whereas in this case, the way that you've set up your experiment, if I understand it correctly, it's the distance matters, but
Yeah.
which direction the error is doesn't. for
Yeah.
example.
Yeah.
Well, everything is hues here, but
Right, but they're still symmetrical. Well, okay.
they're not. But right? I mean, unless you set up the space to be, but that's not what I'm getting to. Unless you set up the space to be perceptually uniform,
Which is how we
which
start.
is hard if you're doing this on mechanical
Which
turf.
I actually think they're doing. So we didn't do that, which is one of the things that we always get in trouble for, but I'm pretty sure that they're doing that.
Oh, are you?
Yeah, because their category sizes are exactly the same. In the real world, they're not. But
No, but I'm saying, but part of the problem, I mean, just, if you're doing this through mechanical turk, you almost can't set.
No, right, but all the monkey stuff is on a calibrated monitor, and when we brought human subjects in and did calibrated monitors and all that, yeah.
Sealed out, right? So it's the presentation, so it's the space that they're sampling the colors from. It's
Right. I mean, that wasn't the point that I was trying to make. It was just assuming this is a perceptually uniform space, whatever.
Yeah.
The distance matters, but the direction of the distance, for example, whether it crosses a category boundary, doesn't matter. That's the point I was trying to
the
make.
category boundaries matter, so I'm not sure I get your question.
Can I change the direction of Milton's question?
Yeah, I mean,
You can impose whatever reward structure you want on either the humans or the monkeys. Right? You could, for example, make the errors have differential costs at different parts of the space or
Yep.
asymmetrically or whatever you wanted. But if that had the influence that you would expect it to have on performance, either for humans or monkeys, that's an argument in favor of the kind of Bayesian model that Canel was talking about, as opposed to the attractor model that you're talking about. So, meaning there's no rationale in the attractor model why the error penalties would have consequences for the neural dynamics.
Unless you're setting up the attractors based on the.
Yeah, on reward,
On
right?
reward.
Yeah.
You
Yeah.
know, if it's supposed to be the operationalization of the Bayesian model, then that would be the rationale. I mean, that's the advantage of a kind of theoretical level, approach like a rational model. Is that there's some basis for, first of all, why the subject's performing the way they do, but also the influence of manipulations make the rewards.
Yeah. Yeah, and I think our goal is just to take those rational models and try to make them more mechanistic. That's the hope, right? So then, yeah, I mean, like obviously, we would predict that one would expect that reward manipulations should change the Bayesian model, and so obviously we'd have to integrate that into our function in some way to account for that. Yeah, absolutely. Yeah.
There, aren't you also assuming that like the cost is being bundled into the perception? Whereas you could imagine like a decision theoretic model that, there's a perceptual module that does the correct perception and then it gets integrated with some cost function later.
No, I'm just asking, how is this being set up? Is this being set up based on, so whatever these categories, wherever these attractors, to use this model, wherever these attractors are, is that based on environmental prevalence, or is that based on costs of errors? Right? Is that based on practical categories?
Yeah.
That's basically my second question.
Right, and I don't think we can distinguish between those two here, right? Like, because they're one and the same.
No, in principle you could, by setting up a different
Yeah, in our current
reward
experiments,
structure in your
yes,
experiment.
sorry.
Yeah.
In our experiments, we can't
Yeah.
differentiate between these two. Yeah, I think that's fair. Okay. All right. Yeah.
Doesn't this differentiate between those two things? You're asking, if I'm misunderstanding the question, maybe, but like it sounds like you're asking, do we have attractors that are like built in because of our natural experience with the natural world?
No,
versus,
no, not just natural experience. Is it a function of just prevalence, or is it a function of decision making under those conditions, right? And the asymmetric costs of making different types of decisions.
Yeah, and I don't know that we can distinguish that here. Yeah. All right. This will take me I can do it pretty quick. I mean, we're close to 3 o'clock. I don't know if people want to hear some monkey data or not.
Please.
Yes.
Okay. All right. So let's go back to deep breath.
Sorry.
No, it's fine. It's fine. It's just we're going to change a little bit here, right? So let's think about our little cartoon model working memory again, right? And we have these Google verification codes, which we want to remember, right? And so we want to hold them in working memory. We want to allow them into working memory. This process is something that's in the literature is referred to as gating something into working memory. And you can gate multiple things into working memory, right? I've just shown you tasks where people do that. And you can also ignore things, right? So if a distracting stimulus comes in, music in the background, for example, you don't want to gate that into working memory until you can close that gate. This is the psychological model, right? And then once you have multiple things in working memory, I just showed you that they interfere with one another, they interact with one another. I mean, this is another thing that we didn't really look at or didn't talk about, but what the exact color identities are, they merge, they attract one another. It's a mess, right? And so that model for sure does not capture all of these things, but at the very least, there's some interference between these items that you can hold in working memory. And then there's a second kind of control process, right, that you could do where you can kind of select one of these items to report, right? And this is a process that we call selection. You can also think about it as kind of like internal attention. We had a recent paper on this, which if you're interested in this, we kind of go into the details and the mechanisms of this. Instead, I want to talk about something new, and so let's talk about kind of this gating process instead. So to look at gating, what we had to do is we had to design a task that monkeys could be gating in or ignoring different stimuli, right? So the monkey would see a stimulus, say this green frog. On this trial, and I'll explain how they know this in a second, on this trial, this is the attractor stimulus, so they should ignore it. And so the gate stays closed, nothing goes into memory, and there's a memory delay where there's no sensory input, nothing's in memory. Then later on in the trial, they might see something that they have to gate into working memory, right? So again, the psychological model says you open up this little gate, and there's this process that moves the sensory information into memory, and then it's the sensory input goes away and the gate closes, but this item is then going to be held in working memory because it's already in there, right? That's the whole point of working memory, is it's kind of bridging. Sensory inputs have been removed, but you're able to still kind of maintain those and operate on those inputs. Then you might get another distracting stimulus, say this ladybug. Now you're still maintaining this memory representation. The sensory input is something you want to ignore, and so you keep the gate closed, and so it doesn't come in and interfere with what is in working memory. And so again, when it goes away, it should disappear. And then in this task, what the monkeys were doing is they were looking for a sensory input that then matches what was in memory, right? So later on in the trial, they'll get a repeat of this stimulus, and they'll say, okay, this is the same thing. And so there's some process in which they have to combine sensory and memory information to make a response. So this really is literally the task that the monkeys did. They saw a sequence of images. Some of them were distractors, which they ignored. Some of them were stimuli that they remembered. more distractors that they ignored, and then a target that they matched. How do they know what to remember? Well, we started off the trial with these little cues, and we had different cues that basically meant if you see something like a blue heart, then if the next stimulus that you see that's either a blue jay or this kind of red and black frog, you should gate into working memory. Okay? If you saw a different cue, then you would remember the frog, the green frog, or the ladybug. And so with this design, each presentation of these stimuli on one trial might be a distractor, and on the next trial could be something that you're supposed to remember. And so everything's nicely balanced. We have multiple cues to make sure that this isn't cue related and blah, blah, blah. Okay, so again, this is just our psychological model of what's going on. And we're going to look at kind of 3 stages of these trials. There could be more than one. There could be 0, or there could be up to 2 of these pre-distractors. Monkey didn't know. There's obviously always something that the animal had to gate. And then again, they would see these kind of what we call post-gating distractors, where again, there could be 0, there could be up to 3 of these on any particular trial, and then the trial always ended with the monkey seeing a repeat. During these pre-distractors and post-distractors, these particular images could 100% repeat, right? So it wasn't that the monkey was just looking for the repeat of any particular stimulus. It had to be this one particular stimulus that was being held in working memory. Monkeys did the task well, way better than I ever thought they would, getting kind of 91 or 97% correct. These are really long time periods, right? So they're remembering these stimuli for up to 7-8 seconds, which for kind of modern working memory studies is a long time. We recorded from 3 brain regions. So we recorded from a sensory region in the temporal cortex called IT, which has kind of form-related representation. So you'd expect nice visual responses to these different objects. We recorded from lateral prefrontal cortex, which of course is the classic area that's thought to be involved in working memory. We recorded 1,000 neurons in lateral prefrontal cortex, and we recorded about 500 neurons in the striatum, with the idea being that these 2 regions, prefrontal cortex and striatum, are maybe involved in controlling this gate. And so really, I'm just going to quickly talk about 2 questions that we've looked at so far. One is how does a stimulus's representation, neural representation, change depending on whether or not you're going to ignore it or you're going to remember it, hold it in working memory. So basically, is it not gated or is it gated? And then the second question we're going to ask is what controls this gate, right? This is our psychological model, which is really nice, but there has to be some physiological basis, presumably, for this gate, and we're curious about what that is. So let's start with our first question.
So how did the monkeys know what cue or stimulus were they given to the gate and store the blue jay by doing it?
Sorry, I went over it quickly. It's this cue here, right? So we had a cue that we presented at the start of the trial. So the monkey saw blue heart, and what the monkey knew through training was that when blue heart is seen, first time I see a blue jay or this red and black, whichever one it is, I need to remember that one, and then look for its repeat. Once I remember, let's say on this trial, the monkey's remembering the blue jay, this could then become a distractor after that. So it wasn't looking for a repeat in one particular set of cues. It really had to update its memory for that particular stimulus. All right. So let's first look at how neurons represent, let's first get this first question. It's how do neurons represent sensory inputs and memories? This is looking at these pre-gating distractors. So this is a stimulus that the monkey should ignore. The stimulus comes on here at 0, and it goes off here at 385 milliseconds later. And then we had this memory delay that lasted out to about 950 milliseconds from the start. The y-axis here is the amount of information that neurons, in this case in IT, are carrying about the identity of the stimulus. So is it a green frog, or red, black frog, ladybug, or a blue jay? And you can see in IT, you get this nice, as we expected, you get this nice sensory information about what the identity of the sensory input is, and it kind of goes away. Maybe there's a little bit of memory, but not much. It's pretty weak. In prefrontal cortex, you get kind of the classic thing, which is you get slightly delayed relative to IT, you get a nice sensory response, and then it just keeps going, right? The monkey's maintaining the memory of what it saw before, even though this is a distractor, right? There's still this kind of short-term memory representation. What happens for the gated stimuli, so when the monkey's supposed to remember it? Again, in IT cortex, you get a nice sensory response. Maybe it's a little bit stronger than the distractor, right? So maybe there's some attentional effect here going on. But by the end of the trial, it's kind of gone, right? There's not really much memory information in IT, in visual cortex. In prefrontal cortex, you get a really much clearer, much stronger response in prefrontal cortex that then is just very nicely maintained throughout. And we can again look at these kind of post-gating distractors, and you get, which is this orange color here, very similar to the pre-gating distractors. IT goes away, prefrontal cortex, a weaker response that's then maintained. So at this point, right, it looks like very similar. In both cases, like, okay, fine, there's stronger representation of the gated stimulus, but you're kind of getting the same response. You're getting the same memory about both of these, both the distractors and the target. And so might be thinking, uh-oh, there's something wrong with our psychological model here, or the monkeys weren't well trained, or something was going on, right? And so we were curious about what is going on here, and so we just said, okay, well, what happens to this memory representation as we look farther in time, right? So the monkeys are seeing the sequence of images, and so we could just look at, like, okay, does this memory last as you present the next stimulus? And so here, what we're looking at is just the information, again, on the y-axis as a function of time on the x-axis. This is just in prefrontal cortex, because that's the area that shows this memory response. And again, if you look at this pre-distractor stimulus, it goes up and it's maintained. There's this like kind of memory representation. Then another input will come in. Could be a gated memory, or it could be a pre-distractor. It actually doesn't affect the results at all. Here I'm just showing the pre-distractor. So here, if we get another distractor coming in, obviously before it's presented, the monkey doesn't have any information about it, right? Because this next stimulus is going to come on here at this 950 milliseconds. So that next stimulus comes on, you get nice information in prefrontal cortex about that new stimulus that just was presented, and you lose the information about what was in short-term memory. So this is like a fragile form of memory. You maintain what's going on for short periods of time, but over time, that any sort of new input that comes in is going to essentially overwrite it. You're going to lose it. So what happens with gating? Well, gating, we can do the same thing. We can look at these gated stimuli and then look at the response to this post-distractor when a new input comes in. The representation of the gated stimulus goes in, stays in. It maybe is decaying slightly as time goes on, but it doesn't really seem like it's overtly affected by the input of the new sensory stimulus. You can get nice decoding of the sensory stimulus, right? That's this blue line. This is just kind of a replication of what I showed you before. It's like you still have this, you have the sensory representation, and then you actually still have a short-term memory representation of the other item. But they don't seem to really interfere with one another. And so we were curious about, okay, what is going on to allow this to happen, right? To allow you to have kind of like a working memory representation, right, which is what's being gated, and then a short-term memory representation of what is like kind of the last sensory stimulus you saw. We've done this in lots of different ways. I'm going to show you this one way in which we've tried to kind of visualize and give you an intuition about what's going on. We were recording 1,000 neurons in prefrontal cortex, right? So you have this 1,000-dimensional vector of neural responses in prefrontal cortex. Monkey is seeing a sensory input, right? And so we can think about that as being encoded in this vector of neural responses. We want to look at what this vector looks like. Of course, we can't look at it because it's a 1,000-dimensional space, which we can't visualize. So what we're going to do is we're going to just project it down into a 2-dimensional space. This is kind of using a variant of principal component analysis, which I'm happy to talk about if people are interested. But basically, think about this as roughly equivalent to 2 orthogonal dimensions within this 1,000-dimensional space that are trying to capture as much variance in the neural activity as we can. And so what this what these 4 different lines here are showing is the presentation of the 4 different stimuli that the monkey saw, starting here at this green dot, which is when the stimulus came on. They move up to this red dot when the stimulus goes off, and then they move back down to this black dot when the memory delay comes on, it ends, right? So that's the end in memory delay. And so if you look at this, right, it's kind of evoking, if you think about this across time, what's going on at the neural population level is you're getting this kind of evoked response that's going up and then coming back down, right? The next distractor stimulus would come in, it would go up and come back down, and go up and come back down, right? It's just doing this little circuit, mostly aligned in this kind of vertical. Now if we look a little bit later in the trial, at when the target comes on, you get a much different response. Right, so now, rather than moving up and down in this vertical, you now move strongly to the right horizontally. We can look again later on the trial at these later distractors, and again now you're getting this vertical response. It's going up, coming back down. And so what we think is going on is that this vertical dimension is basically representing what the short-term memory of what you just saw is. This horizontal dimension, right, which is an orthogonal dimension in this like 1,000-dimensional space, is representing what's being held in working memory. Right? Because they're orthogonal to one another, I can move neural activity up and down along this vertical axis, and it's not going to change this horizontal axis. And that is how we think these representations are being protected from interference, right? I can put in and wiggle this thing up and down all day long. It's not going to change what's being held in working memory. And so we think there's basically two different subspaces there's actually three. So there's a sensory representation in prefrontal cortex that evolves into a short-term memory representation, or it can evolve into this working memory representation. And these things are orthogonal to one another. Then once the target comes on, you move back and the whole thing starts again, right? So like it's, you've essentially circular trials, and so you see circular movements in space. Okay, that's that story. One slide really on how does what controls this gating process. There's sort of two dominant models out there in the literature. One is that prefrontal cortex should be controlling it, and the other is that striatum should be controlling it. And we basically find that both seem to be involved. And so these are two example prefrontal cortex neurons or two striatal neurons that respond differently as a function of whether or not a stimulus is going to be gated into working memory or not. Right? So gated are the blue curves, not gated are the gold curves. The 4 different lines are 4 different stimuli. And again, it seems agnostic, right? Like these neurons are responding the same way if something is gated or not gated, regardless of what the identity of the actual stimulus is. And so these are what we think of as like kind of gating neurons that are kind of maybe acting to open up this gate. And we think what they're doing is that they're acting on the neural representation in a way that's essentially rotating it so that instead of evolving here, it's going to evolve here. That's sort of our ongoing hypothesis, and we're really trying to understand what that means. Like, what is a rotation?
But it looks like the latency in the PFC is longer. And
maybe if you look at the population, it doesn't. We were hopeful. It doesn't really hold up at the population level. They really seem to come on at the same time. Yeah, individual neurons. Like anytime you find a real nice, clean, crisp neuron, it always looks like striatum. And you get these nice things where striatum tends to be up or down, like not gate. This is a not gating preferred neuron. This is a gating preferred neuron. And you get a lot of gating preferred neurons in prefrontal cortex. So there are differences, and we're still trying to tease that out a little bit. Yeah. Okay. Yeah, so just to summarize this, right, we think that in this sort of working memory tasks, there's actually 3 different kind of subspaces. One is just a pure sensory response that will evolve into a short-term memory response, or it can evolve into a working memory response, where the difference between short-term memory and working memory is that short-term memory is fragile. It can be overwritten by new sensory inputs, and what we're calling working memory is kind of robust to that. It's distractor resistant. And we think that this is due to these different kind of subspaces within prefrontal cortex. And that maybe prefrontal cortex and striatum are playing a role in kind of inducing these transformations. All right. Thank you, and I want to say thanks to Pavlos. So Pavlos was the grad student who is now at Google, who did all of the gating stuff, so the second short story I told you. And Matt Panicello was the grad student who's now in Tara Moore's group at Stanford, who did all of the work on working memory and the selection of working memory and the attractor dynamics. Good. Thank you.
I do have a question about the last part. So I think there are two related questions that I have. I was wondering, I don't know whether it's directed to the topic at hand, but what's the advantage of keeping a distractor as soon as you understand that it's a distractor in your short-term memory? That's one thing. And following that, if you made the delay until they see the next distractor longer, would you anticipate that distractor to even fade away further? And could that be explaining why they kind of it seems like they reset right now, but maybe as soon as they understood the distractor, they would just get rid of it?
Yeah, these are two great questions that I don't really have an answer to.
Okay.
I would assume that, yeah, if you made that delay longer, you might lose it. As to why they're keeping it around, so we Alex Libby, she was a grad student in my lab, she did this really nice study where we're recording in auditory cortex of mice listening to sequences of tones. They didn't have any task. They were just hearing really bad music, right? Four notes in a row, over and over and over for 4 days. They probably hated it. You find, right, but this is like we were manipulating like kind of what the first tone, how the first tone predicted like the third tone in the sequence. And it turns out that you see this learning, even though the mouse had no task to do, you see this associative learning of the first tone to the third tone. But the first tone's long gone by then, right? And so you have to have a short-term memory representation of that first tone. And in fact, you can see that even in primary auditory cortex of a mouse doing no task, right? And so, I don't know. Who knows, right? But that's one possible explanation for why you would want to keep these things around. Is that it's just helpful to know, kind of like, what have I seen recently? How does it relate to what I'm seeing now? Are there regularities I can pick up on?
I think in a context where there is no goal, it does make sense, but for this specific context, they know that that's a distractor because they know what thing to keep track of, right? As soon as they decide that it's a distractor. That was my expectation, but I totally understand what you're saying.
Or you could see that they always have multiple goals, regardless of what you as the experimenter want.
Yes.
I mean, maybe, I don't know. That's our assumption about how well they know the expectation of the task.
It's true. So this is not, monkeys have a working memory load of like 2 or 3. And so this is not pushing them. Like if I asked them to remember 3, then maybe this would be higher. And also, right, like they probably could be keeping this information around because they may be a little bit confused about what that cue means, right? Like, should I remember that, right? And so like maybe that's why. Yeah.
I mean, I was thinking the same thing, like why does it stick around? But isn't it also the case that they can possibly repeat, even if the ones you shouldn't remember?
Yeah.
Right? So doesn't that give you an advantage if you sort of let it hang around, then you can faster reject it next time it comes up? A little bit like priming.
Yeah.
I still think it's one of these things that, like, what if I have
Surprise, say run is going to be a test, I better hang on to it.
No. I'm just saying if I have you running in my lab doing a psychophysical experiment, and then the fire alarm goes off, you got to react to it because I think attention's leaky because you constantly have to be aware of it. So I think monkeys are the same way, but people are never just doing one task.
Okay.
That's what I would say.
Yeah.
Sure, but I think the question is still, why would it want to hang around in working memory? I mean, fire alarm, so I can take in new information, but why does it need to stick around if I know it?
It's the leakage of perception. Like you kind of get that image and then you hold on to it.
Well, I think
after image almost.
Maybe this has, so you were saying like what you maintain in working memory is also a function of attention. Right.
It is. Yeah. I mean, the monkeys have to attend to every single stimulus here to discriminate it. So, yeah, we don't really. Actually, that's not entirely true. I sort of left this out, but we had one stimulus that was always. It was like this weird mix of the 4 stimuli, and it was just always a distractor. Monkey completely ignored it, and we still see representations of that. So, yeah, I don't know. I mean, all these theories, I think, could be very true. We don't really have a good way of knowing, other than to say that they do, and that it seems to be somehow more fragile. Yeah.
Chewing on your trajectories in PCA space there, and I'm trying to figure out how to ask my question. I guess the first part is,
Me too.
what about errors? On trials
Yeah.
where the animal doesn't gate things the way it's supposed to, do you see a different shaped manifold in that space?
I really wish we could answer that. We just had not many errors, right? We had like, yeah. So, because if the gating, this also goes into this gating thing. Like, it would be really nice to have some like behavioral correlate of saying like, okay, when the gating, if this is a gating signal in striatum, like when it's good, then like you should see good performance. When it's bad, you see bad performance. We just don't have enough trials to do this sort of thing. But it's a good question.
So I guess the follow on then is, if you actually look at the weights that are composing your principal components, how sort of sparse is that weighting? Do you have really different populations of neurons
Yeah.
in a part of one component versus another? Or is it all sort of multiplex, or what's going
That's
on?
a good question. Yeah. So it's all multiplex. Yeah. Neurons in prefrontal cortex just respond almost randomly. Like if you were to look at a single neuron in prefrontal cortex, in fact, people did this like, I don't know, 10, 15 years ago, and they just say, oh, it's random, right? And I think, because it really does look that way, right? It's like really depends on like whether or not it's early or late, and like what time it is, and how satiated the monkey. I mean, like it changes quite a bit. So this is really something that's kind of a distributed representation. It's not like you get a short-term memory pool and a working memory pool, which would have been nice, but no.
Have you tried this even in piloting with three cues or 3 cue stimulus pairs?
No.
I was just trying to think, because you went with 2, and I was just thinking you definitely have more errors.
Yeah, we definitely would. Yeah. I mean, honestly, I thought this task would be impossible for the monkeys to learn. Yeah, this was surprisingly easy for them to learn.
Surprisingly.
Yeah. So, we just went with the minimum. Sorry. Still took several years to train them. To be fair. I just thought it would take 5, and it took like 2. Yeah.
Quick question. If you have a valid cue, an image after a valid cue, does the IT neurons do they fire with higher amplitude than with an invalid cue?
So we didn't have valid and invalid cues.
Or you cue the animal as to like that purple heart is going to be a blue jay, right?
Yeah.
And so after the purple heart in IT, let's say they see something that doesn't correspond with
Yep.
the purple heart. Does those images have a reduced?
Yeah, just a little. So like that's what this is trying to show here, right? So in IT, there's the blue is something that would be valid, or I think as you're using the term valid, and the yellow would be like a mismatch, right? So it's something they should ignore. And so I don't know to what extent you think these two are different. Yes, maybe there's a slight attention effect here. I think if you do statistical tests, there's a weak effect there, but it's pretty weak.
But then you can go to classic visual neurophysiology when you microstimulate, right, against
Yeah.
the invalid cue, right, to boost up the neurons and to see it out by sort of what's storing
Yeah.
in the future.
Yeah. That's a cool idea. Not something we've done yet. We have, in a totally different task, we have pushed around that selection process. So you can stimulate an FEF and force the monkey to select one item or another, which is cool. So like you can basically control what they're thinking about. Yeah. But no, that's a great idea. So the prediction would be that you kind of boost this up and somehow that like latches it in better into prefrontal cortex, right? So there's classic models of like how delay activity is generated in prefrontal cortex that involve, these sort of bistable kinetics and like say calcium or other. I mean, people have come up with different dynamics that you could use to generate essentially like a saddle point, right, in the neural dynamics, and so the idea would be a stronger input would kind of kick you over that, and you end up in the latch state. Yeah. I don't, at the population level, we don't really see a whole lot of that's consistent with that, because it seems to be a different representation, right? So, yeah.
So I know you're big, or you have been big, on neural synchrony and oscillatory power and phase lags, things like that. Did you build any of that kind of analysis of relationships between overall population level activity and their individual neurons into these analyses?
No, it's on our to-do list. I spent a lot of my early career kind of like looking at how different brain regions synchronize or don't, depending on what the animal's task is. Different populations of neurons become synchronized or don't. To be honest, I feel like I've kind of drifted away from that. Not because I don't believe any of it, or like I feel like that's kind of stagnated. It's like, okay, yeah, we see that, and people are just showing it over and over and over, and what's missing is that sort of like next step of like really understanding. It's been very hard to sort of understand what's generating this synchrony, and so like I think that part of the field, to me at least, seems a little bit stuck against that wall. But I think to your point, right, like could we relate some of those oscillations maybe to these transformations, right? So like we see this rotation basically here, like, yep, that's something we are very excited to try to see. Yeah, because we have good models.
That's what I predict you'd find it.
Yeah, I would love to find that, because so we have some theories about like EI balance and oscillations sort of reflecting changes in EI balance, and as you move to different kind of states of EI balance, you can get different dynamics that might emerge from the system, which is a very hand-wavy way of like, yeah, and we've built some models to show that, but we haven't really looked at our neural data yet.
All right, so I think we're out of time, but thank you so much.
I said I predicted it.